Okay, I'll now be going through question 32 onwards for the 2021 MCQ paper. Okay, which statement correctly compares alcohol and lactate fermentation? A. Alcoholic fermentation produces CO2, but lactate fermentation does not. This is correct. Okay. Next, B. Alcoholic fermentation produces ATP, but lactate fermentation does not. This is not true. Lactate fermentation also produces a bit of ATP. That's the whole point of fermentation for anaerobic respiration. So they can produce a little bit of ATP even if oxygen is not present. Next, C. Both processes can occur in humans. This is not true. Alcoholic fermentation does not happen in humans. D. Both processes produce acetaldehyde. This is only produced during alcoholic fermentation. So D is also not correct. Next, let's go to question 31. An experiment was carried out over 10 minutes to investigate the rate of respiration in living snails. The table shows the average distance moved by the colored liquid towards the boiling tube. So actually this liquid is moving towards the boiling tube. We find that in setup 3, the liquid moves by around 0.5 cm. What is this caused by? Likely caused by CO2 being absorbed by soda lime. Next, we see that the distance in setup 2 moves by around 3.8. What is this caused by? It's not caused by absorption of CO2 into the glass beads. Glass beads can't do anything. Instead, it's likely because oxygen is being absorbed by the living snails because they need it for cellular respiration. At the same time, these living cells will also produce CO2 at the end of cellular respiration. Right? So it's a mixture of O2 being used and CO2 being produced that causes this to go towards the boiling tube. But it also means that more oxygen is being taken in than CO2 being released. Next, we look at setup number 1. Average distance moved by a colored liquid. Okay, in this particular setup number 1, CO2 is being absorbed. At the same time, O2 is being absorbed by the living snails. Any CO2 that's produced by the living snails will go into the soda lime also. So which conclusion can be made from the data? A. Average distance moved by the colored liquid due to absorption of oxygen by living snails is only 4.8. Okay, if number 1 is the result of O2 being absorbed and CO2 being absorbed by the soda lime, and setup 3 is CO2 being absorbed by the soda lime, that means if I take 5.3 minus 0.5, I will find out how much O2 is being absorbed by the living snails, and it's 4.8 cm being absorbed. A is the answer. What about B, C, and D? B. CO2 given out by the snails causes colored liquid to move towards the boiling tube. Okay, it doesn't make sense. If CO2 is being produced, actually the colored liquid should move in the opposite direction, right? More gas, it should push the liquid marker in the other way. So B is not correct. C. Distance moved by the colored liquid in setup 2 is due to the amount of CO2 produced only. Okay, in setup 2, as we said, it cannot be that the production of CO2 caused the colored liquid to move in this direction. For it to move in this direction, some gas must be being absorbed. And so it must also include O2 being absorbed to explain why it moves. So C is wrong. Let's look at D. Setup 2 acts as a control to prove that respiration occurs in living snails. No, setup 2 is not set up to prove that respiration occurs in living snails. I would say that setup 3 is better set up to prove that because they replace the living snails with glass beads. So if there's any gas being absorbed, it has to be from the snails, right? So the glass beads, this particular setup is a better control setup. Okay, so the best answer is A. Let's go to 33, which correctly describes the events happening in the Kelvin cycle. So ADP loses a phosphate group to form ATP. Okay, ADP actually gains a phosphate group to form ATP. It does not lose a phosphate group to form ATP. So this is not correct. NADPH is oxidized to regenerate NADP+. This is correct. When we say something is being oxidized, we mean to say electrons are removed as well as hydrogen is being removed. So this is correct. C. Rubisco catalyzes the formation of CO2 from G3P. This is not true. Okay, in the Kelvin cycle, actually we use CO2 to produce G3P. So this is in the wrong direction, this reaction. D. 3 molecules of CO2 are formed in each cycle. Okay, this is not true. In the Kelvin cycle, CO2 is being used, not being formed. Right? The whole point of Kelvin cycle is to take CO2 from the air to form sugars. So D is not correct. B is the best answer. Let's go to the next question. Question 34. Series of experiments, plants were supplied with water and CO2 containing different oxygen isotopes exposed to light. Okay, what do they mean? They mean to say that the H2O and the CO2 being supplied to the plants, they contain a different kind of oxygen. The oxygen present in the water contains isotope 18 of oxygen. Oxygen in the CO2 is isotope 17 of oxygen. Which statement correctly identifies the products formed in the locations of the chloroplast? So what these experiments are trying to do is trying to track where the water is going. It's trying to track where this CO2 is going by following where oxygen 18 is going and where oxygen 17 is going. So that means wherever we find oxygen 18, it is where the water was used. Wherever we find oxygen 17, it is where CO2 is used. So which statement correctly identifies the products formed and the locations? Stroma, okay, is where the Kelvin cycle takes place, and it is where CO2 is being used to form sugars. So carbohydrate containing oxygen 17. This is correct. A is the answer. Okay, stroma, oxygen containing 18. This is not correct because the water is being used during the light dependent phase inside the thylakoid membrane, right? So not the stroma. What about the thylakoids? Okay, the thylakoids, you should be finding oxygen 18. You should be finding oxygen containing oxygen 18 because we know that the H2O is undergoes photolysis to form oxygen. So any H2O that contains oxygen 18, you should find the oxygen that's produced also contain oxygen 18, not oxygen 17. So actually A is still the best answer. Let's go to 35. The graph shows absorption spectra of 3 different photosynthetic pigments, A, B, and carotenoid. Which statement cannot be concluded from the above graph? Okay, number A, carotenoids can absorb light up to a wavelength of 5 to 5. Okay, where's 5 to 5? 5 to 5 is somewhere over here. So this is true. The carotenoids can absorb light within this range up to 5 to 5. So this can be concluded. Chlorophyll A absorbs light best at 430 and 670. Chlorophyll A absorbs light best at 430. Okay, this is correct. And at 670. This is also correct. So statement B is also correct. Rate of photosynthesis occurs most efficiently from 450 to 500. From 450 to 500. They say this is when photosynthesis occurs most efficiently. Okay, this is not true. Actually occurs most efficiently at 450. Yeah, it occurs most efficiently at 450. If you do want to create a range of some sort, the other range, the other peak is at 670 over here. Alright? It's a bit odd to say that it occurs most efficiently between these two zones. Okay, so this is incorrect and therefore is the answer. D, wavelength of light between 530 is around here, and 600 were least absorbed. And 600 were least absorbed. It is correct. We can see between here to here, the least amount of light is being absorbed. So D is also correct. Okay, so C is the best answer because it cannot be concluded. Let's go to 36. 36 shows you an experimental setup. putting a plant, and then they count the number of bubbles released per minute. Which graph correctly shows the effect of varying the temperature of water on the rate of bubbles produced? Okay, so you expect that. Okay, why are we using temperature? Because we know that photosynthesis also depends on enzymes. And enzyme, the function of enzyme is temperature dependent. We know that enzymes, like any protein, they have an optimal temperature for its function. So we expect that the graph should look something like in T. There should be an optimum temperature, and any temperature outside the optimum, it will not be functioning at its best. You find that before the optimum temperature, the rate of reaction is increasing. That's correct. After the optimum temperature starts to decrease, because as the temperature increases, it starts to denature more and more of the enzymes. So D is the best answer. Let's go to question 37. Question 37 is another Matheson and Starr experiment. They show you two controls, what the DNA band will look like if the DNA is fully heavy or fully light. Then they said that this woman, this student, grew bacteria in 2 generations, first and second generation. Okay, she used DNA that started off with nitrogen 15. Okay, so in generation 0, the DNA was all heavy, right? For the bacteria. In the first generation, however, she made a mistake in her experiment and grew bacteria in the wrong medium for generation 1, which yielded the following result. So what did she grow the bacteria in generation 1? If all the DNA started off heavy and then they became somewhere in the middle band, it means to say that she grew the first generation in nitrogen 14. Why? If all the DNA were once heavy, each one used as a template, if the new DNA that's synthesized used nitrogen 14, which are light, you find that this DNA is half heavy, half light. You'll find them in the middle band. Okay, what about the second generation? We find that in the second generation, the DNA band now, some are heavy, some are in the middle. Likely what happened is this. Okay, each one of these were used as a template. Okay, I'm drawing the template strand now. Okay, so what will make sense for the second generation? Likely, the bacteria were grown in a sample of nitrogen 15. Okay, and any new DNA that's formed is heavy, right? And that will explain why some of the DNA sink to the bottom over here, and why some of the DNA are half heavy, half light, and it's in the middle. Okay, so the best answer is B. If you still don't understand the Matheson and Starr experiment, please go to Lumos and watch me explain how this experiment works. It's very important. Next, let's go to question 38. A diploid cell contains 6 chromosomes. Diploid means a cell that contains 2 sets of chromosomes. Okay, so if the diploid number, 2n, n is a set. If the diploid number is 6, the haploid number will be 3. So the answer is A. It's quite similar to another question in the 2019 paper. Let's go to question 39. Which stage is a part of interphase? Okay, if we look at W over here, we know that this is likely the M phase, because you can see that there are 1, 2, 3, 4 stages. It's likely prophase, metaphase, anaphase, telophase, and then here cytokinesis. So what then is X, Y, and Z? X, Y, and Z combined together is likely interphase. An interphase consists of G1, S phase, and G2. Okay, so D is the answer. Which correctly identifies the stages of the cell cycle where the events are occurring? Okay, so at which stage does DNA quantity increase? We know that during interphase, S phase is where DNA replication occurs. During DNA replication is where you end up having twice the amount of DNA, right? So anything with Y could possibly be the answer. When does cytoplasm quantity increase? That happens during the growth phases. So in this case, X has to be the answer, right? Because it's one of the growth phases. So D has to be the answer. W cannot be the answer. W is referring to the M phase. That's when mitosis or meiosis is occurring. So that's not where cytoplasm quantity increase. All the cytoplasm quantity needs to increase during the growth phases. So D is the best answer. Okay, and that's all for the MCQ for the 2021 paper.